Hello and welcome. This is AQA A-Level Biology. The unit is Organisms Exchange Substances with their Environment. That's Unit 3. Timestamps in the description below if you want to jump between topics, but for now let's get started with gas exchange. Organisms exchange substances with their environment across cell membranes. Surface area to volume ratio can affect exchange of substances. Surface area to volume ratio calculations. We're going to use the example of a cube because it gives us an easy way to do the calculations and show us the impact of surface area to volume ratio. This is a 2 millimeter wide cube. The surface area is going to be the surface area of one side times 6. So it's 2 times 2 times 6 gives a total surface area of 24 millimeters squared. The volume is 2 times 2 times 2, that's length times width times height. That gives a volume of 8 millimeters squared. To do the surface area to volume ratio, we have 24 to 8, that's the surface area and the volume. We can simplify it to 3 to 1, and we can actually simplify it further by dividing 3 by 1 to give us 3. For a larger cube, we have a 40 millimeter cube. The surface area is 40 times 40 times 6, which is 9,600 millimeters squared. And the volume is 40 times 40 times 40, which is 64,000 millimeters cubed. The surface area to volume ratio then is 9,600 to 64,000. If we divide 9,600 by 64,000, we get 0.15 to 1. In biology, we like to do our ratios to 1, and we can simplify that further to 0.15. We could do a couple more examples, but I think this illustrates the idea that the larger the organism, the smaller the surface area to volume ratio. So if we take the example of a single-celled organism, we have a high surface area to volume ratio, and the surface membrane, the exchange surface, is in direct contact with the environment. That means molecules have a short distance to travel to get inside the organism, and the speed of exchange of substances is rapid and sufficient for the organism. The diffusion rate can also be increased further by folding of the surface membrane. So here we have the organism, we now have the folding of the membrane, and so we have a low rate of diffusion on the left and a higher rate of diffusion on the right because of the folding of the membrane. Bigger surface area means a faster rate of diffusion. Larger multicellular organisms have a smaller surface area to volume ratio, and that means diffusion would be too slow to transport sufficient molecules into cells across body surfaces. Most of their cells are too far away from an exchange surface. So if we take an exaggerated example of an elephant, which is a very large organism, it would not be able to take in the substance it needs from its body surface. It would not be able to take in enough substances from its environment across its body surface because its surface area to volume ratio is way too low. A specialized exchange surface and a mass transport system is needed for any multicellular organism. So let's take an overview of the idea of gas exchange. Organisms exchange carbon dioxide and oxygen with their environment. Water vapor is lost from a gas exchange surface as well. So here is our gas exchange surface. This is the outside of the organism. This is the inside of the organism. We often have oxygen diffusing in and carbon dioxide diffusing out, but because the gas exchange surface is exposed to the environment, we have the loss of water vapor as well. So a gas exchange surface should allow efficient gas exchange and minimize water loss. So that's the overall idea of gas exchange surfaces. We're going to take a look at 3 very specific examples, starting off with the gas exchange in insects. It's called the tracheal system. So here is an example of an insect, and it has spiracles, which are small openings that allow diffusion of oxygen. So if we enlarge it there, you can see those little circles on the side of its body. Those are the spiracles. We need to take a look at the adaptation for gas exchange in insects. Here is a top view of our insect. We'll focus in on that area there. We have muscle, we have the tracheal system, and we have the spiracle shown there that allows air to come in and out of the body. We have the trachea, which is this section here, and we have tracheoles, which are these parts here. They are highly branched to give a large surface area. The tracheoles have thin walls, so there is a short diffusion distance for oxygen towards cells, and the tracheal walls are permeable to oxygen. These are some of the adaptations. The abdomen also can expand and contract to cause air movement into and out of the body. So that's how we maximize the efficiency of gas exchange. What about the adaptation to reduce water loss? Well, the insect has an exoskeleton, which is its outer surface layer, made of something called chitin, which is impermeable, so it reduces water loss. We also have spiracles that can close during low active time to reduce water loss. So there we can see our spiracle closing. And finally, we have hairs around the spiracles that also help to reduce water loss as well. Let's take a look at how insects vary the amount of oxygen that's supplied to cells. So we could take another look at our tracheole and see what's going on inside. So here is our working muscle. Here are our tracheoles, and these are our gas exchange surfaces. And this is fluid inside the tracheole. This is mostly water. Let's focus in on that area there and look what happens during high activity times. So during high activity, contracting muscles produce lactic acid. So we can see lactic acid is being produced by the muscle there. This causes a drop in water potential in the muscle. Lactic acid concentration increases in the muscle, and this reduces water potential. Water is drawn out of the tracheole into the muscle. And this means there is more surface available for gas exchange, which is going to be useful for a working muscle. During low activity, contracting muscles produce less lactic acid, so we have a reduction in lactic acid and therefore an increase in water potential. The lactic acid concentration decreases in the muscle, and this increases the water potential. Water is drawn out of the muscle into the tracheole. That means there's less surface available for gas exchange because less oxygen is needed during low activity. Let's take a look at gas exchange in fish. Here we have a fish, and these are the gills, and we can see that the gills are made of gill filaments which increase surface area for gas exchange. If we zoom in on one of these gill filaments, we can see that it has structures on it called lamellae. One by itself would be called a lamella. And we have water flowing across the lamellae. Lamellae increase surface area further. So we have two ways of increasing surface area, but there are further adaptations we need to know about. Lamellae are the site of gas exchange. Let's take a closer look at one. We have blood flowing through the lamella in this direction here, and we have water flowing in the opposite direction. The key features of the lamellae are that they have a thin surface membrane, so the diffusion pathway is short, and a large blood supply to ensure oxygen is taken away and a diffusion gradient is maintained. If we take a closer look still, we can see that blood and water flow in the opposite direction. Water and blood flow in opposite directions. This is called a counter-current system. This maximizes diffusion of water into the blood. If blood and water flowed in the same direction, this is what would happen. This is blood coming from the body, which has low oxygen because it's just giving the oxygen to the body, and we have fresh water coming in with a high oxygen content. Both are flowing in the same direction. If we give an arbitrary value for the concentration of oxygen in the water as 90, in the blood it's 10, which is low. We can see there's going to be a high rate of diffusion of oxygen from the water into the blood. The oxygen in the water reduces and the oxygen in the blood increases, but we get to a point where both concentrations are the same and there is no more diffusion. The concentration in the water and blood would reach equilibrium. No more net diffusion of oxygen into the blood. With the countercurrent system, we have blood flow coming from the body with low oxygen in this direction, and fresh water with its high oxygen content in the opposite direction. So if we look at those arbitrary concentrations again, we've got a high level of oxygen in the fresh water and a low level of oxygen in blood coming from the body. We can see that we always have a diffusion gradient as we move along the lamella. Explaining the counter-current system. Blood from the body and fresh water pass each other in opposite directions. Blood always passes water with a higher oxygen concentration. The diffusion gradient is maintained along the entire length of the gill. This maximizes the amount of diffusion of oxygen into the blood of the fish. Now let's take a look at gas exchange in leaves. Plant leaves exchange carbon dioxide and oxygen with the air. Here is a leaf. We've taken a little slice off the leaf. We have carbon dioxide that can diffuse into the leaf, and we have oxygen that can diffuse out. Their gas exchange surface also loses water to the environment. Water loss needs to be balanced with efficient gas exchange. Let's zoom in on that area there, and this is a leaf cross-section which has been magnified. We have what's called mesophyll cells, palisade mesophyll along the top and spongy mesophyll in the middle area there, and these provide the gas exchange surface. We can see that water and oxygen can diffuse out of the leaf, and carbon dioxide can diffuse into the leaf for photosynthesis. What are the adaptations for gas exchange? Well, leaves are thin to provide a short diffusion distance for gases, and leaves are usually wide and flat to provide a large surface area for gas exchange and for light absorption. Mesophyll cells are loosely packed and provide a large surface area and allow easy diffusion of gases into and out of the leaf. Stomata can open and close to allow gases to enter and leave the leaf. Adaptations to control water loss. This is for leaves. Continued. There's a waxy cuticle that reduces evaporation of water. So on our diagram there, it's the waterproof layer on the top. The stomata can close to reduce or stop water evaporation. So here's an open stomata and here's a closed stomata. Stomata are mainly on the lower surface, reducing direct exposure to the sun and therefore reducing evaporation. Let's take a look at something called adaptation in xerophytes. These are plants that are adapted to dry conditions. Xerophytes are plants adapted to dry conditions. Example number one, a very common example, are cacti. That's the plural for cactus. We have a thick waxy cuticle to reduce water loss from evaporation. This is a thicker than average cuticle. We have a thick stem to store the water, and we have a wide and deep root system. It's wide to collect water before it evaporates in the warm sun, and it's deep to collect water from far underground. Stomata only open at night when it's a little bit cooler, so there is reduced evaporation of water. Leaves are also thin spines to reduce surface area and to reduce water loss. Here's one of the leaves. It's a very thin spine and it's designed to reduce water loss. Our next example is something called marram grass. Marram grass is found on sandy beaches. Water drains rapidly from sand and wind speeds are usually high on beaches, which speed up transpiration. So here is an example of some marram grass. We could take part of the structure of the grass there and we can see it's like a rolled up structure. Leaves are rolled up. The trapped humid air inside the rolled up leaf reduces the evaporation rate of water. However, if we zoom in there, or if we magnify there, should we say, we have the inner surface of the leaf and we can see that the air in there is very humid, and this humid air has a high concentration of water particles, so it reduces evaporation from the leaf. We have hairs. Hairs reduce air movement and trap humid air, which reduces evaporation. The thick waxy cuticle reduces evaporation from the outer surface of the leaf. We also have a wide and deep root system which allows water to be collected more efficiently. The stomata there are in what we call pits. That means they're sunken in, which means no air movement in those pits and high humidity, which reduces evaporation further. So lots of different adaptations in marram grass and cacti to help reduce water loss, but also to allow gas exchange as well. Let's take a look at the human gas exchange system, the lungs. Here are a pair of lungs. We can open up one of them so we can see inside. Here we have the trachea, which brings air into the lungs. Here we have two branches off the trachea called bronchi. One is called a bronchus, two are called bronchi. And the bronchi branch off into bronchioles. At the end of the bronchioles, we have structures called alveoli. So here is one alveolus. The alveolus has many capillaries surrounding it. Many capillaries transport oxygen away and maintain a steep concentration gradient of oxygen. Each alveolus has a large surface area, and there are millions of alveoli. Let's take a simple look at an alveolus. We have fresh air coming in, and the epithelium is the wall of the alveolus. The epithelium is only one cell thick, and we can see it's pointed out there. And if we magnify this area here, we can see that we have the epithelium wall there, but we have the wall of the capillary there, which is called the endothelium, and that is also only one cell thick. This provides a short diffusion distance for gas exchange. CO2 can very quickly diffuse out, and oxygen can very quickly diffuse in. Ventilation in the lungs. Ventilation moves air into the lungs and maintains a concentration gradient of gases for rapid diffusion. Here is a simple diagram of the lungs. We have the bottom part there that's called a diaphragm. That's a flat sheet of muscle. We have muscles called intercostal muscles, and those are found between the ribs. If we just take a more detailed look of the ribs, this is a side view. Here we've got two ribs, and in between them are the intercostal muscles. The internal intercostal muscles are shown there, and the external intercostal muscles are shown there. Let's see how these structures help with breathing. First, we'll deal with breathing in. The external intercostal muscles contract and the internal intercostal muscles relax. The rib cage is pulled up and out. The diaphragm contracts and flattens, and the volume of the lungs increases and pressure in the lungs decreases. The pressure becomes higher outside than inside the lungs. Air moves in. That's breathing in. Breathing out. The external intercostal muscles relaxed and the internal intercostal muscles contract. As a result, the rib cage is pulled down and in, and the diaphragm relaxes and moves up. The volume of the lungs decreases and pressure in the lungs increases. The pressure becomes higher inside than outside the lungs. Air moves out. Next, we're going to take a look at digestion and absorption. Digestion and absorption. During digestion, large biological molecules are hydrolyzed to smaller molecules that can be absorbed across cell membranes and into the body. Here is a digestive system. Food enters and is hydrolyzed into small molecules. Anything that cannot be digested passes out the other end. So here we have our large molecules. They undergo hydrolysis to make smaller molecules. If we were to magnify the small intestine or the inside of the small intestine, we could see that there are cells that line the small intestine, and this is where the small molecules are absorbed into the blood. So we have large biological molecules that undergo hydrolysis to make small biological molecules, and these can be absorbed into the blood. Once they've been absorbed into the blood, they undergo mass transport around the body to the places where they're needed. So let's take a look at the large molecules and the small molecules that we're talking about. The first example is protein for large molecules. These are broken into short polypeptides, and these short polypeptides are broken down into our small molecules, which are our amino acids, which can be absorbed into the blood. For carbohydrates, those are broken down into disaccharides, and those can be broken down into monosaccharides, which can be absorbed. For our lipids, they are broken down into monoglycerides, fatty acids, and glycerol. Carbohydrate digestion. Here is a part of the digestive system. For carbohydrates, we have the enzyme amylase, which is produced in the mouth by the salivary glands and is produced by the pancreas as well. We also have membrane-bound disaccharidases embedded on the membranes of cells in the small intestine. So we can see those enzymes there. Let's take a look at the digestion of starch first. Here is part of a starch molecule. Here's our enzyme amylase, and we can see how that works. Starch is hydrolyzed by amylase into maltose, which is a disaccharide. Digestion of disaccharides. Here are some examples of some disaccharides. Here are our membrane-bound disaccaridases. And we can see that these are broken down into monosaccharides. Disaccharides are hydrolyzed by membrane-bound disaccharidase enzymes into monosaccharides, and that's summarized in the diagram above. Protein digestion. Proteins are made of polypeptide chains, which are made of amino acids. So here's part of our digestive system again, and the enzymes are endopeptidase and exopeptidase enzymes in the stomach and small intestine. We also have membrane-bound dipeptidase enzymes in the small intestine as well. So how does endopeptidase work? This hydrolyzes peptide bonds within a polypeptide. Exopeptidase, these hydrolyze peptide bonds at the end of polypeptides. So if I've got a long polypeptide here, we can see the action of an endopeptidase that digests within the polypeptide molecule. So we can summarize that like this. This produces short polypeptides. Exopeptidase enzymes can work on short or long polypeptides, but this is how they work. They hydrolyze at the ends of polypeptides, and that will end up with the production of amino acids. The last part is dipeptides and how they're broken down. These are hydrolyzed by membrane-bound dipeptidase enzymes, as you can see there in the membrane. Let's just take a look at one example of a dipeptide being hydrolyzed. There we go. But these will then be hydrolyzed into amino acids. Let's take a look at the digestion of lipids. Here's part of the digestive system. We have the liver, which produces bile. We have the gallbladder, which stores bile, and bile during lipid digestion passes into the small intestine. Bile has two roles. One, it neutralizes stomach acid, and two, it emulsifies the lipids or the fats to form small droplets. Here are our large droplets of lipid. They have a low surface area to volume ratio. We can go through emulsification to make small lipid droplets which have a higher surface area to volume ratio. So let's take a look at one of these droplets and magnify it. We can see it contains triglycerides, and those are broken down into different parts. They're broken down into monoglycerides and two fatty acid chains. The next stage is slightly complex, but let's go through it. Here is a cell lining the small intestine. It's called an epithelial cell. First stage is that monoglycerides, fatty acids, and bile form structures called micelles. Here are some micelles. The micelles travel to cells lining the small intestine. There they go. And the monoglycerides and fatty acids are absorbed by diffusion into the cell. The micelles are not absorbed. It's the fatty acids and the monoglycerides that are absorbed. A very important point there. Triglycerides are then reformed in the cell and form structures called chylomicrons. The chylomicrons are packaged into vesicles. There's our vesicles containing the chylomicrons. And these are released into the lymphatic system and eventually into the blood. So that's the details of the digestion and absorption of lipids. Next, let's take a look at mass transport. Mass transport. Exchange surfaces provide gases and nutrients for an organism. The nutrients are transported around the organism, and this movement to where they are needed is called mass transport. Mass transport in animals. Let's take a look at hemoglobin. Hemoglobins. These are a group of chemically similar proteins. They are found in various organisms. Hemoglobins are proteins and have a quaternary structure. This means there are 4 polypeptides joined together. Each polypeptide contains an iron ion. Hemoglobins are found in red blood cells. Red blood cells have a large surface area for more absorption of oxygen, and they have no nucleus. This gives more room for hemoglobin. Hemoglobins transport oxygen. One hemoglobin molecule can carry up to 4 oxygen molecules. Hemoglobin in red blood cells transports oxygen from the lungs via the heart to respiring cells. Here's our lungs, here's our heart, and here are our respiring cells. Oxygen is transported from the lungs to the heart to our respiring cells. Transport of oxygen. Hemoglobin has different affinity or attraction for oxygen depending on its environment. Hemoglobin has a high affinity for oxygen where levels are high, for example in the lungs. In this case, hemoglobin loads oxygen. In other words, it picks up oxygen. Hemoglobin has a low affinity for oxygen where levels are low, for example near respiring cells. So hemoglobin unloads oxygen. We can summarize this in a diagram. Hemoglobin becomes oxyhemoglobin by loading or associating. That's in the lungs. And oxyhemoglobin can undergo unloading or dissociating to become hemoglobin. That means to unload or get rid of or lose the oxygen. This happens near respiring cells. The cooperative nature of oxygen binding. Let's simplify our hemoglobin molecule up here and see what happens. This is hemoglobin. Here are some oxygen molecules. The first oxygen molecule does not bind easily with hemoglobin. Once it binds, the hemoglobin changes shape. This makes it easier for further binding of oxygen. So we can look at that in the form of a graph. On our graph, on the x-axis, we have something called the partial pressure of oxygen, measured in kilopascals. And really all we need to know about that is that is an indication of the concentration of oxygen in the environment. Next is the percentage saturation of hemoglobin with oxygen. That's an indication of how much oxygen the hemoglobin is carrying. We call that percentage saturation. At first, we know that it's hard for the first oxygen to bind, so percent saturation increases slowly. However, once the first oxygen molecule has bound, oxygen molecules bind more easily. So we have a steep increase in the graph. Towards the end, we have a reduced number of binding sites left for oxygen, so the graph tails off a little bit. The graph is less steep. So let's take a look to see how this is useful. I'm going to use the color green to represent oxygen. We have our lungs, and here, when we breathe in fresh air, we have a high ppO2. That means a high partial pressure of oxygen, a high level of oxygen in the air. We have a low concentration of CO2. Our red blood cell here can represent our hemoglobin, and hemoglobin has a high affinity for oxygen in the lungs because oxygen levels are high. Oxygen is loaded into the hemoglobin. The oxygen-rich blood travels to cells. These are our respiring cells where there is a low partial pressure of oxygen because the oxygen is being used up. We also have a high carbon dioxide concentration. Hemoglobin here has a low affinity for oxygen, so oxygen is unloaded into the respiring cells. That's very useful because the respiring cells require the oxygen for aerobic respiration. The Bohr effect. Here's our graph again showing the oxygen dissociation curve. This is the one that we drew just now. If we take an example of a partial pressure of oxygen of 8 kPa, we can see the saturation is about 60%. But if we have carbon dioxide in the blood, this increases the acidity of the blood, and that has the effect of decreasing the affinity of hemoglobin for oxygen. In other words, the hemoglobin will hold on to the oxygen less. The high CO2 concentration makes the curve shift to the right. So if we take the same concentration of 8, we can see there's a much lower saturation of hemoglobin with oxygen. For the same partial pressure, hemoglobin holds less oxygen and it is unloaded into respiring cells. There it goes. The hemoglobin holds less oxygen for the same partial pressure when there is carbon dioxide around. This is called the Bohr effect, and the curve is towards the right. Let's take a look at an example of other hemoglobin. Fetal hemoglobin, that's hemoglobin in unborn babies. Here's our curve as before, our normal curve that we saw. For fetal hemoglobin, this has a higher affinity or attraction for oxygen than the mother's hemoglobin. The oxygen dissociation curve for fetal hemoglobin is to the left of the mother's. So for example, at a partial pressure of 6 kPa, the mother's hemoglobin is around about 31% saturation, whereas the fetal hemoglobin is closer to 78% saturation. The result is, for the same partial pressure, fetal hemoglobin has a higher saturation of oxygen. This means the fetus will take oxygen from the mother's hemoglobin. Let's take a look at some other oxygen dissociation curves that have come up in past papers. Mice have high metabolism. Here's our usual oxygen dissociation curve. Their cells need more energy. The oxygen dissociation curve is to the right. Hemoglobin unloads oxygen more easily. More oxygen is supplied to cells for faster aerobic respiration. And this is because mice have a high metabolism, so lots of aerobic respiration is required. Lugworms, which live in burrows. Here's an example of a lugworm in a burrow. Water in the burrow has low oxygen partial pressure. The oxygen dissociation curve for the lugworm is to the left. This gives a high oxygen saturation at low partial pressure. So here's our low partial pressure, and we can see there is a high percentage saturation of hemoglobin with oxygen at that low partial pressure. This means more oxygen is absorbed from the water. Llamas, they live at high altitude. The air has a low partial pressure of oxygen. So the oxygen dissociation curve is to the left for llamas. This gives a high oxygen saturation at low partial pressure, similar to our lugworm. More oxygen is loaded into hemoglobin in the lungs at low partial pressure. Next, we're going to take a look at blood circulation in mammals. Here's the circulation system, or a simple version of the circulation system, with the parts that we need to know about. This is the right side, and this is the left side. Remember, when you're looking at it on paper or on a screen, left and right are the opposite way around. This represents the lungs, where gas exchange happens. This section down here represents the whole body, including the head. This is where gas exchange happens as well. This is our heart, and this represents oxygenated blood, and this color represents deoxygenated blood. This is our aorta. This carries oxygenated blood to the body. This is representing the renal artery that carries blood to the kidney, so that area in the middle there is the kidney. This is the renal vein, comes from the kidney. This is the vena cava, which carries deoxygenated blood from the body to the heart. This is the pulmonary artery carrying deoxygenated blood to the lungs. This is the pulmonary vein carrying oxygenated blood from the lungs to the heart. Let's take a look at the flow of blood around the body. So if we start at the left ventricle there, we're going to label the heart in a moment, we can see the blood travels in this direction here. So to the body, back to the heart, then to the lungs, and back to the heart again. This is called a double circulation. So that's the inside of the heart, but if we look at the heart from the outside, a couple of important labels here. These are the coronary arteries in that dark red colour. These supply the heart with oxygen and glucose for respiration, and so therefore muscle contraction. Let's take a look at the labels for the human heart. Remember, left and right are opposite when we're looking on a screen or paper. So here's the heart. We start off with the left atrium. Here we have the atrioventricular valve. It's called the bicuspid. And this allows the flow of blood in this direction. We could just zoom in there a little bit, just so we are clear about the position of the atrioventricular valves. They're circled in white. We have then the left ventricle. This is the right atrium. This is another atrioventricular valve. On the right hand side, this is called the tricuspid. And this allows blood to flow in this direction shown here. And then we have the right ventricle. This is the aorta, this is the pulmonary artery, this is the pulmonary vein, and the vena cava. We also have semilunar valves in the aorta and pulmonary artery, and these allow the flow of blood in this direction. So they are circled there in white. So these are the key labels for the heart. Let's take a look at the cardiac cycle. Here is the heart. We start off with blood entering the two atria. It enters at the same time. Pressure slowly builds due to the entrance of blood into the atria. We then have the atrial walls which contract. This increases the pressure in the atria. That means the pressure is higher in the atria than the ventricles. The atrioventricular valves open and blood flows into the ventricles. Then we have the ventricle walls which contract. This increases pressure. The pressure is higher in the ventricles than the atria and the arteries. The atrioventricular valves close and the semilunar valves open. Blood flows into the aorta and the pulmonary artery. The valves stop the backwards flow of blood. Let's take a look at the structure and function of arteries and veins. Here's our simplified version of the circulatory system again. Here are our arteries, and arteries carry blood at high pressure. Here's our veins, and these carry blood at low pressure. The left ventricle pushes blood at high pressure to the body through the arteries. Arteries need to withstand high pressure and maintain high pressure. All arteries carry oxygenated blood except the pulmonary artery. There's our pulmonary artery there. And pressure reduces by the time the blood reaches veins. Veins do not need to withstand high pressure. All veins carry deoxygenated blood except the pulmonary vein. There's the pulmonary vein there. Arteries. These always carry blood away from the heart under high pressure. Here is a cross-section of an artery. It has a thick muscular wall, it has a thick elastic wall, and it has a small lumen. That's the inside part of the artery. Just on the inside there is a layer called the endothelium. Thick muscular walls contract to maintain pressure. Thick elastic walls stretch and recoil to maintain pressure. And the endothelium, which is the inner surface, is smooth to reduce friction and reduce chances of clotting. Veins. These always carry blood towards the heart. They have a thin elastic wall, a large lumen, and thin muscle walls. The endothelium is again just on the inside. Veins carry blood at low pressure. Blood flow is helped by the contraction of muscles. So there's a cross-section of a vein, and muscles contract and help to push the blood forward. Valves prevent the backflow of blood. So blood cannot flow backwards. Capillaries. Arteries branch off to make smaller blood vessels called arterioles, and these branch off into capillaries. Capillaries are the site of exchange. Here's a capillary, very small, it's about 10 micrometers wide, and it is only made of a wall called the endothelium. It has a very narrow lumen and permeable walls which allow the movement of gases but not large molecules. Red blood cells pass in single file to increase surface in contact with the wall. This causes or allows faster diffusion. This also decreases blood flow to increase the time for diffusion to happen. The endothelium is only one cell thick. This gives a short diffusion distance. There are many capillaries which increase surface area, and this ensures all cells are close to a capillary as well. Formation of tissue fluid. We're going to deal with just this topic on this one slide. So here is a person, and here are some cells from that person. This section here is called the capillary bed, and it's important as an exchange surface. This is the artery end. Blood from the heart comes into the artery end, and that's at high pressure. This is the vein end. Blood back to the heart at the vein end. This is at low pressure. This is the capillary. These are our body cells, and this is something called a lymph vessel. And this yellow area here is tissue fluid. So at the left-hand side, we have high hydrostatic pressure, and this becomes low hydrostatic pressure as we travel across the capillary bed. So let's see what's happening. At position number 1, we have high hydrostatic pressure due to the ventricle contraction in the heart. This forces water and dissolved solutes, for example glucose, out of the capillary into the capillary bed. This liquid that has been forced out is called tissue fluid. The tissue fluid contains nutrients and oxygen for the cells. Point number 2, large structures, for example red blood cells, and large molecules, for example proteins, remain in the capillary. 3. Loss of water from the capillary decreases the water potential of the blood. At point 4, water enters the blood in the capillary by osmosis. Point 5, excess tissue fluid is collected by a lymph vessel and eventually is returned to the blood. Next, let's take a look at mass transport in plants. Mass transport in plants. Here is our plant. We have xylem tissue that transports water and minerals up the plant by the transpiration stream. The transpiration stream is the movement of water through the plant. Transpiration, however, is something slightly different. This is the evaporation of water from the leaves. Transpiration stream and transpiration are not the same thing. If we take a detailed look at the stem, we can see that we have xylem vessels in there, and water and minerals travel upwards through the xylem vessels. These are tubes made of elongated dead cells. They also have rings of lignin for support. If we take a closer look on the inside, we can see there are no end walls of those long cells. This is so water can pass through. Cohesion tension theory. This explains the movement of water up the plant through the xylem. First step, water evaporates from the leaves. This draws water from the xylem. This creates tension, which is a pulling force on the water. Water is pulled up the xylem as a column, and cohesion between water molecules allows an unbroken column of water to rise up the xylem. The lignin rings stop the xylem vessels from collapsing. If you're asked to explain cohesion tension theory, that's going to get you the marks. Translocation and mass flow. Phloem tissue transports organic substances, for example sucrose, around the plant. This is called translocation. Here's our plant again. We have structures called companion cells and sieve tubes. Phloem tissue. This is made of elongated tubes of living cells. End walls, called sieve plates, have pores to allow dissolved substances to pass. There are no nuclei and limited organelles, which allows the flow of dissolved substances. Companion cells help with the process of translocation. The mass flow hypothesis. Here are our companion cells, and we have the leaves, which we call the source, produce glucose, which is converted to sucrose. Sucrose is actively transported into phloem cells by companion cells. This lowers the water potential in the phloem, and water enters from the xylem by osmosis. This produces higher hydrostatic pressure in the phloem, which means higher fluid pressure. This means the mass flow of sucrose solution to respiring cells or storage tissue, for example, roots or seeds. The storage tissue is called the sink. Sucrose is unloaded from the phloem by active transport into the sink. This increases the water potential in the phloem, and water leaves the phloem and enters the xylem by osmosis. This causes a fall in hydrostatic pressure. So we can see we have a difference in hydrostatic pressure between the source and the sink, and this is what is thought to drive the flow of solution around the plant in translocation. Use of tracers and ringing experiments. If you've got this far through the video, well done. This is the final part. Ringing experiments. A ring of bark is removed from a woody stem. The ring of bark removed contains phloem. This removes the phloem but not the xylem. So there's no phloem in that section there. After a period of time, a bulge appears, and this contains sucrose solution. There is no bulge in the section below. Any non-photosynthetic tissues below the ring die. The conclusion: translocation of sucrose occurs in the phloem and not the xylem. Sucrose produced in the leaves is transported downwards via the phloem. Death of tissue below the ring indicates no sucrose is transported to them. Finally, tracer experiments. Here's a plant. Here is a transparent bag over the leaves of the plant, and this bag contains carbon dioxide with radioactive carbon, called carbon-14. Any compound with the radioactive carbon can be detected with X-ray film. So the carbon-14 in the carbon dioxide gets incorporated into glucose and into sucrose. If we take a knife and cut a cross-section of our stem, this is our stem cross-section. We have phloem on the outside and xylem on the inside there, as shown by those labels. And here is some X-ray film. This turns from white to black when exposed to radiation. We can place the film on our stem, and we can see the areas where there was phloem. It contains the radioactive sucrose. Radioactive carbon is also detected in other regions of the plant, for example, in the roots. This is the sink. This provides evidence for the movement of carbohydrates in a plant. So that's a summary of Unit 3 for AQA A-Level Biology. Remember, do check your spec and do have a go at practice questions, but this is just an overview of what is in Unit 3. Thanks for watching, and I'll see you soon.